Hello students, in this video we are going to discuss about a very important topic, that is enzymes. Enzymes can be defined as these are the complex nitrogenous organic compounds which are produced by living plants and animals, which catalyze the biochemical reactions in the living organism. So such complex nitrogenous organic compounds are called as enzymes. Since it is produced by living plants and the animals, and it plays a very important role in the living organisms. So different reactions are taking place in the living organism. So it have a very important role in all these biochemical reactions. So they are actually proteins. So we can say that chemically all the enzymes are the proteins. Also, these are called as biocatalysts because the numerous reactions that occurs in the bodies of animals and plants to maintain the life processes are catalyzed by the enzymes. Thus, we can term the enzyme as biocatalyst. And the phenomena is known as a biochemical catalysis. Without the enzymes, living process would be very slow. So, the enzymes have very important role in the living processes. They are actually the protein molecules, therefore they have high molecular mass and they form colloidal solution in water. When we look at the example of enzymes, which plays a very important role in the different reactions which taking place in the body, like inversion of sugar. Sucrose with water, when it undergoes hydrolysis, it gives rise to glucose plus fructose. This process is called as inversion of sugar. The invertase enzyme converts the cane sugar into glucose and fructose. So the enzyme here, invertase, is actually plays a very important role here because it catalyzes the reaction of hydrolysis of sugar into glucose plus fructose. The next is the conversion of glucose into ethyl alcohol. So the enzyme here, that is zymase. The enzyme zymase convert the glucose into ethyl alcohol, that is also called as ethanol. This reaction is catalyzed by the enzyme, that is zymase, and carbon dioxide is the side product which is formed here. Next is the conversion of starch into maltose. Starch and maltose both are the carbohydrate. So the conversion of starch into maltose, it takes place with the help of an enzyme called as diastase. So the enzyme diastase convert the starch into maltose. That is the importance of this enzyme. The next one is the conversion of maltose into glucose. So maltose can be converted into glucose, that is the hydrolysis of maltose will takes place with the help of enzyme called as maltase, and we get here glucose as a product. The next is decomposition of urea, that is into ammonia and the carbon dioxide. So the enzyme called as urease catalyzes this decomposition. So here the urea, formula for urea is NH2CONH2. So on the decomposition, it gives rise to ammonia and carbon dioxide. So this reaction is catalyzed by the enzyme called as urease. So these are the certain reactions we have seen in which the enzymes have an important role to catalyze these reactions. And what we can see from these, that is in the tabular form, we can see that the enzyme invertase, and it is found in the yeast, zymase is found in the yeast, diastase in the malt, like this. So here these enzymes convert a particle reactant into the particle product. That means we can say that enzymes are specific in nature. That means a specific enzyme can catalyze a specific reaction, that is a biochemical reaction. So the properties of the characteristic features of enzymes are in this way. It is highly efficient. It means one molecule of an enzyme may transform 1 million molecules of reactant per minute. That means the enzymes are quite efficient because a small quantity can catalyze a large quantity of reactant. So they are highly efficient and they are highly specific in nature because each enzyme is specific for a specific reaction. That means one catalyst cannot catalyze more than one reaction. That means it is highly specific. So that is, we have seen that for the specific reaction, specific enzymes catalyzes. So we can use the property here, that is, it is highly specific in nature. Next, they are highly active under the optimum temperature. That is, the rate of an enzyme reaction become maximum at a definite temperature. That is called as a optimum temperature. So the activity of enzyme is highest at a temperature which is called as optimum temperature. On the other side, the optimum temperature, the enzyme activity decreases on the other side of optimum temperature. That means that they work more efficiently, active under this certain temperature. That is the body temperature, human body temperature. That is the temperature ranges between 298 to 310 Kelvin. So the optimum temperature ranges for the enzymatic activity is the ranging between these. So the human body temperature being 310 Kelvin, it is suited for the enzyme catalyzed reactions. So what we can say that the activity of enzyme is high during these temperature. Next, they are highly active under the optimum pH. So the rate of the enzyme catalyzed reaction is maximum at a particular pH, and this pH is called as the optimum pH. That means the range between 5 to 7 pH, we can say that the enzyme are more active. That means the rate of enzyme catalyzed reaction is maximum at this particular pH, which is called as optimum pH. The next point is increasing the activity in the presence of activators and coenzymes. So the enzyme activity is increased in the presence of certain substances which is called as coenzymes. And it has been observed that when a small non-protein is present with an enzyme, that is non-protein part like vitamin, if it is present with an enzyme, the catalytic activity is enhanced, or we can say it increased. So due to the presence of coenzymes or the activators with the enzymes, the activity will be increased, such as the activators like metal ions, etc. It increases the activity of enzymes. The next is decreasing the activity in the presence of inhibitors and poisons. Like ordinary catalyst, enzymes can also be inhibited, or we can say it is poisoned. That is by the presence of certain substances. These are called as inhibitors or the poisons. So these actually inhibit the catalytic action. So that is, it decreases the activity. So the enzyme activity get decreased in the presence of inhibitors and the poisons. So the use of many drugs is related to their action as enzyme inhibitors in the body. So different drugs we are using, that is actually inhibiting the, or we can say it is controlling the function of enzymes. So that is actually different drug are there. So that is actually the mechanism we'll be discussing in the chapter Chemistry in Everyday Life. So these are the important characteristic features of enzyme catalysis. Now let us discuss the mechanism of enzyme catalysis. So before explaining the mechanism, let us discuss what is active site. The enzymes have specific sites on its surface where the substrates get bind and the catalytic activity takes place at this site and having a characteristic shape and possesses certain groups. So what is actually this active site? As we have discussed about enzymes, and the role of enzyme is actually to accelerate the rate of the reaction. So here the enzymes have certain specific sites that you can see here. This is an enzyme and it have a specific site on the surface of it. That site is called as active site and which have actually a characteristic shape and it possesses some active groups like amino group, acidic group, OH group, etc. So these possesses active groups and the site is called as active site which have a particle shape also. So whatever the substrate, that means actually the reactant. These are called as actually the reactant. Reactants, when it approaches to the enzymes, it binds on this site. That is a site which is found on the surface of the enzyme. So the substrate get bind on the site and which have a characteristic shape and possesses some active groups. Such sites are called as active site. Now we'll be discussing the mechanism of the enzyme catalysis. So here the enzyme, we are using the letter E for the enzyme, S for the substrate. So what actually takes place, the substrate gets bind with the enzyme and it makes a complex, or we can say an intermediate is formed, that is called as enzyme substrate complex. And finally, it will break up into enzyme and the product. So that means the substrate which get bind on the enzyme, here the formation of products takes place at the active site and the product get removed. So at last the enzyme which was as a catalyst which was after the reaction, you can see that the catalyst or that the enzyme is as it is. There is no change in the enzyme. So that is what we can say, that the enzymes are the catalyst which plays a very important role in the reaction, but at the end it will be remain as it is. Chemically, there will be no changes takes place in the enzyme and the product is formed here. So this can be explained by the mechanism that is called as lock and key model. As we know that a particle key can only open the particle lock. So similarly, it is considered, the mechanism of enzyme catalysis is considered with the lock and key. Just like a particle key can only open the particle, or it get inserted into this hole of the lock. That is in the same way we can say that the reactant molecules, that is called a substrate here, which have a complementary shape that you can see, so that it can fit into the cavities that is present on the surface of the enzyme, that is called as active site. So the substrate have a complementary shape which fit into these cavities just like a key fit into the particle lock. So on the account of this, we can say that the substrate get binded on the enzyme on this active site and it forms a complex which is called as enzyme substrate complex, and the product is formed here and the product and the enzyme get released. So this is what the mechanism of enzyme is considered with lock and key model. The next model is induced fit theory or hand in glove model. As we know that the shape of the glove and the hand is not complementary, but when it is inserted into the glove at the time, we can see that now it get correctly fitted into the glove. Similarly, you can consider as the hand as a substrate and enzyme as a glove. When we look at in the case of enzymes, the shape of the active site of the enzyme and the substrate are not complementary. When you check here, it is not complementary. But when the substrate approaches the enzyme, then slightly changes takes place at the shape at the active site. So what happened here? Now because it is get changed, now the substrate can easily get bind on this site. So what is actually it is taking place? When the substrate is approaching, so the enzyme it slightly changes the shape at the active site so that the substrate can fit into the active site and it forms an enzyme substrate complex. So this model says that it is not complementary, the shape is not complementary, but it get induced to get complementary, and that is called as induced fit theory. So this results in the formation of enzyme substrate complex which get break up into the product and the enzyme. So these are the very most important points related to enzyme. So this is all about this video. Have a nice day.